Hello again. So in the 1998 document, hang on, it's not supposed to happen. In the 1998 document, they mention here that they completed all the major goals in its current 5-year plan from 1993 to 1998. So now they start to not just read the sequence, but now they need to understand the sequence. They focus again, saying that they need the thought of doing whole genome science now is becoming a reality. Okay, then with the availability of sequences, then there is this new, then there's the new need to look at genetic variation. So availability of human genome sequence presents unique scientific opportunities, chief among them the study of natural genetic variation in humans. Basically what they're saying is that they want to look at the variations, which ones are important for diseases, which ones are just polymorphisms. So they need to understand the relationship between genetic variation and disease risks for perhaps use in terms of prevention and treatment of illnesses. Of course, with this new technology and information comes the real issues in the ethical and legal aspects. So the new focus on genetic variation, now that they're looking now not just on the sequence, they're looking at variation as well as other applications, raises additional ethical, legal, and social issues that need to be anticipated, considered, and resolved. This table 1, looks at the some of the achievements that's been made. For example, a genetic map averaged 2 to 5 centimorgan resolution. Now they actually achieved 1 centimorgan, published in September 1994. Physical map, they estimated that they need 30,000, but they actually have 52,000 STSs. So they have achieved the original goals that's been set, but and also they've achieved more than that. So here, human sequence variation analysis was not a goal initially, but now they are mapping the 100,000 map SNPs, single nucleotide polymorphisms. Single nucleotide polymorphisms usually refer to sequences that are within the coding sequence that has bearings or importance in function. And later this document looks into the details. If you want to go into, this is the human DNA sequence. They want to finish the complete human by the end of human genome sequence by end of 2003. Finish at least 1/3 by 2001 because they want to come up with a working draft. They want to at least cover 90% of the genome by the end of 2001. So these are again some of the goals that have been mentioned previously, and they're just reiterating it now. Make the sequence totally and freely accessible. This is very important because there were efforts to to commercialize whatever information that they obtain, but the whole idea of this was to actually provide all information freely. For sequence technology, again, continue increasing the throughput and reduce the costs. Support research of novel technologies that can lead to significant improvements. Later we will see that this is on the NGS. Develop effective methods and advanced development of new sequencing technologies. Goal 3 for sequence variation: develop technologies for rapid, large-scale identification and scoring of SNPs. and other sequence variations. So now it's not just about getting the sequence, they want to develop methods for detecting variations and identify common variants in the coding region of the majority of identified genes, and then create a map for these variations, develop the intellectual foundations for studies of sequence variation, and then create public resources of DNA samples and cell lines for other people who's interested to do the same kind of work. The rest of the document, again, support research methods for studying function of non-protein coding sequences. Even back then, it was already acknowledged that there are sequences within the non-coding that is important. The numerous sequences responsible for other functions such as control of gene expression, RNA splicing, formation of chromatin domains, maintenance of chromosome structure, recombination or replication, and all this rely on the non-coding sequences that are being transcribed. So they need support, and well, by what do you mean support? That means funding as well, to enhance research on that area, develop technology for comprehensive analysis of gene expression. So you can see the shift from figuring out how to sequence, and then figuring out how to sequence faster, and then now once you have the sequence, you need to make sense of it. Now, with also the information from other model organisms, there is a new field called comparative genomics. That means you compare between the structure of different organisms from different species. So for that, they need to complete the sequence of C. elegans, complete the sequence of Drosophila and the mouse. And of course, to do that, you just need to go through the same processes. You need to develop physical and genetic mapping resources, cDNA, and then complete the sequence, and so on. All right. And because they are successful in identifying they're successful in sequencing the first few organisms. So they said, now identify other model organisms that can make major contributions to the understanding of the human genome, and this include the pufferfish, some other organisms later that they thought will be very, very useful. So again, number 6 is looking at the ethical and legal and social implications with now the increased knowledge of the structure and the functions of the human genome. So you need to be prepared with what are the ethical questions that's going to arise. Bioinformatics, again, improve the content and utility of databases, develop better tools for data generation, capture, and annotation. So now it's about annotation. Annotation is like putting information, develop and improve tools for databases for comprehensive functional studies, develop and improve tools for representing and analyzing sequence similarities and variation, create mechanism to support effective approaches for producing robust, exportable software that can be widely shared. Okay, so I think that's about it on reading this documents. If you look into Spectrum, there are other documents. For example, this is the review that was some of the review frequently asked questions about the Human Genome Project. You can have a look at that, and then these are some of the again summary of what it was. It says yesterday and today, but this was actually published in 2010. So still, it was about almost a decade ago. All right. So I'll see you in the next document. Bye bye.